Kia ora koutou. Ko Tiffany taku ingoa, he kaireruku tau whainga aho ki Manaki Whenua. Good morning everyone, my name is Tiffany and I am the Events Coordinator at Manaki Whenua Land Care Research. A lot of you would have joined us previously for a webinar session, but before I hand over to Christine, I'm going to run through a couple of technical slides to ensure that your experience with us online today goes as smooth as possible. If you are new to our LINKOnline sessions, bear with me while I give you a quick intro to our webinar platform. You will notice you have a control panel at the side of your screen. If at any time this collapses, you can bring it back by simply clicking the orange arrow button. If you are having sound issues and you can see my mouth moving but cannot hear a word I'm saying, please grab the PDF in the handout section, and this has instructions to resolve this quickly. The audio panel is where you can control where the sound plays on your computer. Select the drop-down arrow and choose your audio output. During the presentation, you may have questions that you want to be covered in our Q&A session. You can do this via the chat panel throughout our session today. You will notice it is pretty small and it will be hard to read other attendees' questions. Select the pop-out icon on this panel and drag the corner out to make it as big as you want. You can also use this feature if you are having technical issues and ask me any questions. Questions asked by the audience show as anonymous and a green colour in the chat panel. However, please note we will use your name in the Q&A session. If I respond to you regarding a question, this will show as red. Now over to Christine to introduce you to our 7th LINKOnline session.
Kia ora, and thanks Tiffany for getting us underway so successfully. I'm just here to introduce Jamie very quickly, and we'll hand over to him. His presentation will take about 20 minutes or so, and so we've allowed a few minutes at the end, maybe about 10, for some questions and answers. So my request of you is that you put your questions into the chat box as early as possible. That improves your chance of getting a live answer, but just to let you know that we will address all questions even if we don't get to them online today. So you know, even if it's something that we're not going to have time for, please put your question in there. We really are interested to know. So I'm keen to introduce Jamie Wood. He is a member of our Long-term Ecology research group, and amongst other things, he manages our ancient DNA laboratory. Over to you, Jamie.
Thanks everyone for joining this webinar today. As Christine said, my name's Jamie Wood and I'm a paleobiologist with the Long-term Ecology research group here at Lincoln. In our work, we get to travel back in time 50,000 years or so to study how animals, plants and other organisms have responded to major environmental perturbations in the past, whether those be changes in climate or whether they be anthropogenic influences. And today I'm going to talk a little bit about the work that our group has been doing over the past 10 years or so, specifically that which relates to conservation. But before I begin, I'll just point out that we do have a website and a Twitter feed where we post updates and information about our work. So if anyone's interested in learning a bit more detail about what we do and the different methods that we use in our work, I'd encourage you to go and check these out. Now, prehistory might not seem particularly relevant to current day conservation management, but in this presentation, I'll aim to prove that the past does have a role to play, specifically around informing different aspects of threatened species conservation and ecosystem restoration. The idea of conservation paleobiology has been around for quite a while. But I guess it's really gained international momentum over the past 10 years or so. And during this time, there have been a number of discussion papers devoted to the topic. The field of conservation paleobiology was really born out of the recognition that by looking back into the past, we can gain unique insights into a number of different aspects of species and ecosystem biology that are relevant to conservation. Now, these insights might complement observations made on modern ecosystems, or they might be completely unique insights that would not have been possible to obtain by studying modern species in current ecosystems. In this presentation, I'll talk about several of these in more detail, specifically ecological surrogacy, so how introduced species might replace extinct ones within ecosystems. I'll talk a bit about ecological interactions between species and investigate baseline states. We all know that globally ecosystems have gone through large amounts of change in the recent past. Species populations have declined or become fragmented. Habitat has been lost. Ecological interactions between species have been broken. And knowing something about how these systems were prior to the disturbance can help guide their restoration and management. New Zealand is no different in this regard. So we have experienced large losses of native habitat types, such as forests and wetlands, and many of our native species are now restricted to small, isolated populations compared to their former extent. Yet, compared to a lot of other land masses around the world, New Zealand's past is still so recent and it has a lot of relevance to current day ecosystems. Think, for example, of some of our oldest living trees, whose branches might once have held the nest of our extinct hawk, or grown from a seed dispersed by a giant moa. Here in New Zealand, we're in the somewhat fortunate position that we have excellent records of our past species and ecosystems. For example, we have widespread organic soils and peats, and these preserve pollen grains and spores, which we can use as a record of vegetation changes through time. In these same soils and peats, we can find charcoal fragments that provide valuable histories of fire frequencies and histories of fires that can be studied alongside these vegetation changes. These soils also contain other proxies, a wide range of things we can look at. For example, testate amoebae, which can provide clues as to things like hydrological changes in the past. In New Zealand, we also have some of the richest subfossil deposits in the world, such as this cave where the surface is covered with scattered moa bones. And these deposits preserve valuable records of prehistoric fauna in the form of bones, but also things like DNA, which can be extracted from the sediments and be used to reconstruct the animals and plants that once lived around these sites. So what sort of questions might we be able to explore using these types of biological archive, and what relevance might the answers have for conservation and restoration? The first case study I'll present today shows how paleobiology might allow us to compare species that never co-occurred in the same place at the same time and establish whether a recently arrived species, in this case deer, are performing a similar ecological role to that of an extinct taxa, in this case the moa. New Zealand forests evolved alongside large herbivorous animals, 9 species of different moa, which ranged in size from 15 to 250 kilograms in weight. Following the extinction of moa, there were several hundred years where large herbivores were absent from New Zealand forests, and this time is commonly referred to as the herbivore gap. The herbivore gap came to an end following the introduction of ungulate herbivores such as deer in the late 19th century. Now, this has caused 2 hypotheses to be generated about the state of New Zealand forests. The first is that New Zealand forest understories were largely kept open by moa browsing. Following the extinction of moa, the forest understories flourished, and the thick undergrowth that greeted the first European botanists was therefore kind of an unnatural situation compared to what it had been in the past. The second hypothesis is that deer offered an ecological replacement or surrogate for moa, and they effectively recreated these open, natural New Zealand forests. So how might we tackle some of these hypotheses using our paleobiological techniques? Well, the first thing we need is a little bit of luck, and in this case, it happened to be the discovery of several moa skeletons by trampers in the Dark River Valley back in 2006. I joined an expedition which was led by Otago Museum at the time to the area where these bones had been found, and we ended up discovering a dozen well-preserved moa skeletons beneath these large boulders that were scattered throughout the forest. But perhaps most importantly was that we also discovered alongside one of these skeletons hundreds of well-preserved moa droppings, or coprolites. And radiocarbon dating of these showed that they had been deposited fairly recently, so the oldest were about 1,000 years old, and the youngest were only about 600 years old, or about the time that moa became extinct. However, what was also really exciting was that alongside these moa droppings in the same rock shelter were deer droppings. And this particular piece of forest was quite remote and had remained free from other disturbances such as fires, which might have dramatically altered the plant composition in the past. And so by comparing the content of these moa droppings with the deer droppings from the same site, we could for the first time directly compare the forest composition and the foods available during the time of moa and deer. So our first step in the study was to sequence DNA from the moa droppings. And by doing that, we were able to identify droppings from 4 different moa species that once lived at this site. The next thing we did was extract and analyze pollen grains from the moa and the deer droppings and compare those to each other. And these are the results of that analysis. So in this graph, each row represents a single dropping, which we analysed. And these are identified on the left of the graph here by the species they represent. Those in the white area at the top are the different moa droppings, and those at the bottom in the grey area are the deer droppings. The bars simply represent an abundance or percentage of pollen grains in the sample from different plant types. So don't worry too much here about the individual plant taxa at the top, but what I really want you to take away here is the message that overall difference between the moa and the deer droppings. The moa droppings contain much higher abundance of pollen from sub-canopy trees. So these are things that are occurring in the forest understory, while the deer droppings were dominated by pollen from the beech canopy and spores of ground ferns. If we look at other plant groups, the lianes and small herbaceous ground cover, we can see that these are more common and diverse in the moa droppings compared to the deer droppings. And so what we interpret from this is that the degree of moa herbivory pressure on forest understories was substantially less than that which is experienced under a regime with deer. Many of the plant species that occur in the moa droppings are rare or completely absent from the deer droppings. But in fact, if you go to the site today, you can still find many of these plant taxa that were once eaten by moa. But you have to look pretty hard and you have to climb on top of these big boulders where they've got these little refugial perched gardens that have a high abundance of different plant species that you just don't find down on the forest floor where deer can access them. In fact, most of the forest is now quite bare. It contains just moss and with tall canopy trees in between. And so here we have a case where paleobiology has helped resolve a debate around the degree of the effects of an introduced species, which could have potential implications for management of that species. A slightly different approach to addressing the same question was looking directly for changes in vegetation that coincided with the extinction of moa. And in this study, what we did was we took 3 soil cores from forests that were considered to be unmodified, that is, they hadn't experienced large fires or any other disturbance over the past 1,000 years or so. We then used radiocarbon dating of these soil cores to pinpoint the exact time when moa extinction happened within the core. And then we looked for changes in the relative abundance of different pollen grains across this horizon. However, what we found was that apart from slight increases in Coprosma pollen within the herbivore gap at all sites, we found no evidence for consistent large-scale changes in forest understories following moa extinction. And again, this suggests that the effect of moa was quite minimal on forest composition compared to that, the effects of deer. In the second case study, I'll show how paleobiology might help inform us about ecological interactions between species that could well be essential for one of the species' survival. This is a photograph showing the Honeycomb Hill caves near Karamea, and these caves are very well known for their deposits of moa bones, and you might see a few scattered down in the front of the photo here. But the caves also contain significant deposits of Kakupo droppings or Kakupo coprolites. Many of the plant species that occur in this area now no longer co-occur with remaining Kakupo populations. And so studying these ancient dung might provide potentially useful insights into the suitable food species for Kakupo and the suitability of different habitat types for the reintroduction of Kakupo in the future. So in 2010, we began analysis of Kakupo coprolites from the Honeycomb Hill cave system. And one of the really interesting results we had when we analysed the pollen from these coprolites was the high abundance of a very distinctive pollen grain. This particular pollen grain comes from a plant called woodrose or Dactylanthus taylorii. And it's a subterranean parasite that attaches to the roots of forest trees. Dactylanthus produces nectar-rich flowers which emerge through the forest floor in autumn. Now, unfortunately, these are very attractive to introduce mammals such as possum and rats, which eat the flowers whole. This is one of the key reasons that Dactylanthus is now highly threatened and is reduced to just 4% of its natural range. And translocations of these plants to offshore islands that are free of introduced mammals is one option and has been suggested as a possible management tool. However, pollination of the flowers is also considered a key limiting factor to the future growth of Dactylanthus populations. And this is because the only known pollinator of Dactylanthus flowers are New Zealand short-tailed bats, which in themselves are now quite rare. These bats spend a lot of time foraging on the forest floor, and their furry little faces act as the ideal mechanisms for transferring pollen between plants. But what implications might the discovery of Dactylanthus pollen in a Kakupo dropping have for conservation of the plant? Although parrots can often be destructive feeders, they can also lap up nectar from flowers. Think about an example of kaka lapping nectar from flax flowers. So it's possible that Kakupo and perhaps other birds might once have acted as pollinators for Dactylanthus. One thing to consider is that the behaviour of native nectar-feeding birds has been changed a lot by introduced predators. So these birds spend much less time feeding on the ground than they do in the absence of predatory mammals. Combined with the rarity of Dactylanthus, and it's not perhaps surprising that nobody has yet observed native birds feeding and pollinating this plant. However, paleobiology in this case, the use of Kakupo coprolites, has raised the idea that as a distinct possibility, birds could do this and will serve to direct future study into these interactions. Ultimately, this could have important implications for conservation management of Dactylanthus. And if birds are shown to be important pollinators of this plant, then it opens up a large number of potential new islands where translocations could occur. And these islands may not have thriving bat populations, but they could have really quite substantial native bird populations that could act as pollinators. In the final case study, I'll look at how we can use the past to understand natural vegetation communities. And some of the results might even challenge our assumptions about what is natural in a particular area. Now, this photograph shows Pōhutukawa forest growing among old lava flows on Rangitoto Island. This is a scene typical of offshore islands around New Zealand's northern coast, where forest tends to be dominated by angiosperm species such as Pōhutukawa. The Poor Knights Islands are no exception, and they're currently covered by Pōhutukawa forest. In 2014, our team analysed an 85 centimetre soil core from Tawhirirahi, the largest island in the Poor Knights group. The core spanned a period of 2,000 years, and we analysed pollen grains and ancient DNA preserved in this soil. And it revealed quite a fascinating and unexpected history of vegetation change on the island. So rather than having naturally been covered in Pōhutukawa forest, the results demonstrated that the natural vegetation of the islands was in fact quite different, and it was forests that had been dominated by podocarps, particularly rimu, the nikau palm, and a rich understory of diverse shrubs and ferns, perhaps not too dissimilar to the type of forest seen in this photo. The Pōhutukawa forest that's there now had actually only regenerated since gardening ceased on the island around 180 years ago. And it's likely that given sufficient time, the forest will actually revert back to that original podocarp and palm dominance. Moreover, this scenario might not be unique to the Poor Knights and could represent the situation on a wider number of offshore islands around northern New Zealand. So the paleoecological data has provided a clearer understanding of the history and baseline states for these forest ecosystems, as well as their potential future trajectories, which could help inform decisions around the management and restoration of northern offshore islands. The use of paleobiological techniques to establish which plant and animals were living in particular areas in the past has a wide range of possible applications. And one that's attracted a lot of attention and interest from end users is the potential to help guide local native replanting schemes. And this is especially true in regions where there might be very little native vegetation remaining. And so the only records about what plant species once grew in these areas could be the pollen grains, DNA, or other fragmentary plant remains now preserved in soils, sediments, and peats. And actually, there's quite a good coverage of such data from across New Zealand, and these are just the pollen records that are currently held at Manaki Whenua. Several years ago, we were granted TIFBIS funding to undertake a scoping exercise around developing an online portal, which we called Cravens, that would allow end users to access existing paleovegetation data and help them interpret local vegetation histories. As part of this, we ran a survey of a wide range of potential end users, from educators through to conservation managers, and the majority of which regarded the tool such as this would be very useful for their own line of work and interests. And so we believe there's clearly a need and an interest out there for such information. And our team is currently working towards developing this tool, which we hope will be made available sometime in the near future. And so to finish up, I'd just like to return to this paper, which laid out a vision for the emerging field of conservation paleobiology. I think the authors summed it up quite well with their statement that a crucial challenge in the immediate future is making conservation paleobiology results policy relevant. And I think that here in New Zealand, we've made a really good start towards this, but it's also fair to say that there's a lot more scope and exciting potential for collaboration between paleobiologists, conservation managers, and policymakers going forward. And with that, I'd like to thank you all for listening and welcome any questions.
Hey, thanks very much for that, Jamie. That was really interesting. Questions are rolling in, and the first one is, do we find ancient DNA in our disturbed or intensified farmed soils?
Yeah, it's a good question. It's quite possible there is some there. However, the sites we tend to look for ancient DNA in are sites where we can minimize the contamination from modern DNA. So sites that might have plants growing there today, some of that DNA can contaminate our results. So we tend to look for things like cave deposits. A lot of work's done in permafrost sites where that DNA is kind of frozen in, and sites such as shallow soils could well have ancient DNA in there, but the amount of modern DNA is just going to be really difficult to separate those signals. So yeah, there's still a lot of work to be done on how long DNA exists in different types of environments. So yeah, at the moment we don't tend to focus on those sites in our research.
Great, maybe an opportunity for the future.
Yep.
Now we've got another question here. It's got three bits in it, but I'm going to go for the last bit. Is there any evidence for changes in forest composition following deforestation associated with early human settlement? Obviously, you looked at what Moa had deforested.
Yeah,
we look at the more modern version.
We certainly see recurring patterns across New Zealand and in a lot of our pollen records that we have. So a lot of our cores will extend back 2,000 years or more. And so they encompass that time when people arrived and started modifying the landscape. And so what we tend to see in a lot of the drier eastern areas that were a lot more prone to fire is that there was a period, perhaps of a couple of decades, where fires were quite widespread, and this removed a lot of forest from places like the Canterbury Plains, for example. In a lot of those areas, the forest didn't come back, but in some of them it did. And it'll be really interesting, and I think it's an area of future research is actually to study how similar those forests that came back are to the ones that were there in the past.
Great, thank you. Here's a very practical question. Do you have a favourite example of how your work has resulted in an actual on-the-ground change being implemented?
It's a really good question. From our particular work, we're still working towards that and getting that implemented. There are examples of other situations though where the past has helped inform and actually resulted in practical management, and a lot of these come from overseas, but they are very rare examples. And so I think as this kind of last slide implies, is we've got all this amazing data and we can kind of see how it would be useful, but it's just now kind of implementing that and seeing it through to actually being used in practical ways. And I think that's where we'd really like to go in the next few years.
We've got time for one last question, which really appeals to me. Tom Harding is wondering how on earth you identify pollen grains in 1,000-year-old poo.
Yeah, amazingly they're really well preserved, and so with the Moa poo, it's basically dried out, and so we rehydrate it just by soaking it in a solution for a couple of weeks to soften it up. And then we can just use standard pollen processing techniques in the lab, which involves a number of steps. But we end up with microscope slides that pretty much contain pure pollen grains. And so we just look at those under a microscope, and because they're so well preserved, we can compare them to living plants today, the pollen that we find in those, and get pretty good identification of them.
Fantastic. That's a great way to finish. I can see there are some more questions that have come in, and rest assured we will answer those questions. both directly to the people who have asked. Thank you very much for that. And also by posting a Q&A list on our website along with the recording of the seminar in a week or so's time. So I'd like to say thanks especially to Jamie. Thanks also to Tiffany for making this all happen. And we'll look forward to seeing you all again before too long with another fabulous LINKOnline webinar. Thanks and bye for now.